Hello guys, and welcome to another episode of biology. And of course, today we're going to be looking at Dihybrid Cross. Dihybrid Cross. All right, and also in today's class, we're going to also talk about another law of Mendel, which is the law of independent assortment. In the previous class, we looked at monohybrid cross, looked at law of dominance, looked at a law of segregation, okay? And of course, we did a lot and enjoyed ourselves. If you have not watched that video, you have no business here, okay? Please go find the video on any of our channels, YouTube, Facebook, TikTok. Watch it, then come back to join us so that you can fully understand this class. Do not forget that these tutorials, anyway, these episodes, all of these lectures are brought to you for free, all right? We have heard that people are asking for payment for these videos, okay? Somebody will say, send me 10,000 and I'll send you the videos, all right? It's a lie. We do not sell these videos, all right? They are free. They are free for everyone who's preparing for exam, any O-level exam, JAMB, WAEC, NECO, GCE, NABTEB, okay? They are all free. Please do not be deceived by anyone, all right? So, it's the only thing we sell, okay, that you pay for is the activation of the O3 Schools app, all right? So if you need the O3 School app, you need to pay for the activation, all right? And the activation fee is rightly written on the application, all right? So we'll talk about the features of the app in a moment. Let us see a question from the JAMB app. This is a 2022 question. 2022 question number 17. All right, it says here that in a dihybrid cross, the parents A says carry different pair of alleles for each trait. B says carry same pair of alleles for each trait. C says carry different pair of alleles for all traits. D says carry same pair of alleles for all traits. Okay, this question and many more we are going to answer at the end of the class. Okay, so when we are done with the hybrid cross, any question that has to do with the hybrid cross, okay, we use the question search feature from the O3 Schools application to search for questions, and then we'll answer them together, all right? So please ensure you download the O3 Schools application. Download it, okay, activate it, and start using it immediately to prepare for exam. The app has everything you need to succeed in your exam. All the past questions for all the subjects are on the application, right? What's more, question search feature, you can search for question according to topic, and the question that JAMB, any question that JAMB has said concern that topic, so long you search for it using the question search feature, will come out for you to answer, building your confidence whenever you finish a particular topic. What's more, classroom feature that has latest JAMB syllabus, has lecture notes, video lessons on the app, study plan that can help you to create a timetable. Many of you say, please, Master T, help me to create a timetable that I will use to write my exam. You don't even need our app because of the study plan on the classroom feature. Click on study plan. It will create a timetable for you to crash all the 4 subjects you want to do, all right? All the syllabus, all the topics, and create a timetable for you to tell you, it will be telling you what to do each day. If you say you want to prepare for exam in 1 month, it will crash, it will prepare everything for you so that you finish the syllabus. Many of you will prepare for your JAMB without finishing your syllabus. Okay, that is why you are not performing well. Start early, prepare, finish your syllabus, you will see how good, okay, your results will come out. Also, there's a mock challenge on the application every Saturday. All right, so all students that are writing JAMB, WAEC, or any O-level exam come to the application to compete with each other, all right, to win a lot of prizes. But of course, as you know, the reason why we conduct that challenge is not because of the prizes, is to bring out that spark in you, okay, to get rid of exam fear, okay, because if you have participated in our mock exam, which are exactly like JAMB's exam, all right, definitely you will see that you have nothing to be scared of, all right? You get it to JAMB past question, JAMB likely exam question, okay, and the WAEC app also, WAEC past question, WAEC likely exam question, okay, you will see, you familiarize yourself with how JAMB or WAEC set question, building your confidence, you come the booga, all right? So all of these features that you are seeing right on my left and on my right, all right, goes for a small activation fee, which is less than the price of rice and chicken in a good restaurant. So please do not go and be stingy to yourself, okay, and to your future, all right? You say it too cost. And I'll be laughing on my own, all right? So please get the activation activated. You have nothing to be scared of. If you want to activate your app, message this number on WhatsApp 091-2151-5246, or click on activate on the application. Once you download it, at the top, you find where they wrote buy activation key. Click on the buy activation key. You will see by transfer. Click on by transfer. You see that the correct account number and the correct amount for the activation on the application. Make your payment, send the receipt to this number on WhatsApp. All right, within 30 minutes you will get activation key. And if for example you do not get it within 5 minutes or 10 minutes, okay, know that it's because we are busy in the studio like this preparing classes for you. All right, so now before we talk about the Dihybrid Cross, okay, I saw some questions that related to what's it called, that related to Monohybrid Cross. Okay, calculation question that has to do probability and the rest. Okay, we did not actually solve anyone in the last class, so let's just take one or two and then we'll begin. There's a question here, 1995 number 44. Okay, it's been displayed on the screen. It said the F1 of a cross. Okay, the F1, first filial generation of a cross between a tall and a dwarf plant. Okay, the F1 of a cross between a tall and a dwarf plant was tall. Okay, so after they crossed it, okay, after they crossed the F1, all right, the cross gave us what? A tall plant. Now they say the F1 was advanced to F2, all right? So, okay, so let us self-pollinate what the F1, okay, and see what we are going to get. They say how many of 120 F2 plants will be dwarf? Okay, how many of 120 F2 plants will be dwarf? Out of 120 F2 plants, how many will be dwarf, right? So from the question from the beginning, it says the F1 of a cross between a tall, okay, so a tall, okay, if you are not told, if the question does not tell you heterozygous or homozygous, it does not tell you homozygous tall or heterozygous tall, assume homozygous tall. Okay, it did not tell you heterozygous dwarf or homozygous dwarf, assume homozygous dwarf. All right, so that means the genotype for what? For the tall plant, okay, will be TT, while the genotype for the what? For the dwarf plant will be what? Will be TT. Okay, so now they will what? They will cross. Okay, all right, we'll take one garment from here. This one will bring one garment, this one will bring one. All right, so now we are going to have what now? T, capital T, yeah, all right, then T, yeah, all right, so I'm going to have what? TT, okay, so now it is this F1 that advanced to what? To F2, all right, so now when this advanced to F2, okay, that means we are crossing this against what? This, all right, so this against this is what we are crossing. So if you use our Punnett square, all right, what do you discover? You discover what that here will be T, here will be T, here will be T, here will be T. T times T is TT, T times T is what? TT. T times T is what? TT, T times T is what? Is TT. Okay? So now our result here is giving us what now? How many tall? The phenotype ratio, phenotype ratio here will be giving us how many tall plants. This one is tall, but yes, TT is tall. This one is tall, right? This one is still tall because this capital T is dominant over this recessive what? T. Okay, remember this is what? Recessive. Okay, and then this is what? Is dominant. Isn't it? Okay, so now this one is tall, this one is tall, this one is also what? Tall. Okay, then this one is what? Is dwarf. That means among the possible outcomes, okay, we have 4 possible outcomes. Okay, 3 are what? Tall. Then 1 is what? Is dwarf. Okay, so now they're asking us that if there are 120 F2 plants in total, okay, how many will be dwarf? Okay, so now from this possibility, from this ratio, we will not, for simple mathematics, we did the ratio. Okay, that's why, especially those of you that are doing biology, all right, take your math lessons serious too, all right? You need them. Do not throw away any knowledge you have learned. Okay, so knowledge of ratio is coming to play. Knowledge of probability will be coming to play also. So you cannot throw away any knowledge you have learned in your mathematics. And that is why most times for most universities, during their post-term screening, all right, they also include mathematics in your exams, okay, for medical students, for biological sciences, all right? So because they know that you need knowledge, they use that as an avenue to screen you out. That is why you must be vigilant, okay? So now, total ratio here now, okay, what will be 4? How did I get 4? That is what? 3 plus what? 1. Okay, let me say that is 3 plus what? 1, which is what now? 4. Okay. Now, out of this 4, only 1 is dwarf. Okay. So now, if there are 120 F2 plants in total, that means the ratio of dwarf, which is 1, divided by the total ratio, which is what? Is 4, okay, times 120 total F2 plants will be what? The number of what? Dwarf F2 plants that we have. Okay, so if you look here, 4 here will go 1, 4 here will go what? 30. Okay, so 30 times 1 is what? Is 30. So this means that what? A total of what? 30 plants were what? Were dwarf. All right, among the 120 what is it called now? F2 plants, 30 were dwarf. All right, how many were tall now? All right, obviously, if you want to know the ones that were tall, just simply subtract what? 30 from 120. So if you subtract 30 from 120, you are going to have what? 90. All right, that means our answer here is what? Is 30, because they were only asking us how many plants were dwarf or will be dwarf. All right, that's the question they asked us. Me, I'm just trying to tell you that, let's say you were asked how many were tall among the 120, just simply subscribe. You already know that since 30 were dwarf and 3 were all tall, that means the remaining would be for the tall ones, isn't it? As simple as ABC. Please do not throw away your knowledge of mathematics. Let us see another question. Okay. This is let me see. Okay, now this is 1994. Okay, the first one we saw was 1995, number 44. Okay, was 1995, number 44. Okay, now this one is this one is 1999, number 43. Okay, 1999, number 43. It says that if the offspring of a cross between a brown mouse, okay, given as BB, and a black mouse, given as what? BB, capital B. Okay, that means this one is what? Dominant. Okay, because the capital one is dominant. It's the question that gave us this, so then this one is what? Is recessive. Okay, they say if the offspring of a cross between what? Between a brown mouse and a black mouse are allowed to interbreed, how many different genotypes would result? How many different genotypes? You must understand question. Another thing again, you must understand questions that is being asked of you. Because if you do not understand, you can fail it. All right. So now, if you do our cross, here now we have what? B, one gamete, this one will bring 1, this one will bring 1. If the cross are going to have what now? BB, isn't it? Now, it is this one now that crossed to F2. So I'm going to have BB and what? And BB. Okay, this is what we want to cross. Okay, if you use a Punnett square, let's say we are not using Punnett square, let us go directly. We are keeping this B constant now. We are assuming as if we are not seeing this B now. So I'm going to have BB. Yeah. This B will cross this one also. We also have what now? Capital letter B and small letter B. Then this small letter B will cross this one, big letter B here. Okay, let us do it this way. So I'm going to have B and B. This time now, we're using this small b now, we're not seeing this B. We're keeping this capital letter B constant and crossing with this small b. That's why we're saying B times what? B, BB. Then this times this. Okay? B times what? B is what? BB. All right? So now, they say how many different genotypes would result? How many different genotypes would result? Genotype, not phenotype. Phenotype talks about what? The physical observable features. Okay, what we are seeing on the outside. What will be seen on the outside as a human being is this one is this one black or brown? This one is what? Is black, Abi, because B is what? Dominant. All right. So this one is what on the outside will be seen. This one as what? As black. We'll see this one. This one is definitely what? Black, Abi, because what is BB, okay, is dominant. Then we're going to see this one as what? As black also, Abi. Then this one will not see it as what? As brown. This is what will be seen outside. But genotype, all right, is different, okay? So now, the phenotype ratio, yeah, that they did not ask us all this one. I'm trying to make you understand on a larger scale, all right? So phenotype ratio, yeah, will be what? Will be 3 ratio what? 1, all right? We're having 3 black and what? 1 brown. But the question did not say phenotype. They say how many different genotypes are we going to see? All right? We're going to see what? Black. We're going to see what? Homozygous black. Homozygous. Okay, this one not homozygous, it's homozygous. That is what? This BB here is homozygous black. Abi, this one is what? Is it heterozygous or homozygous? It's heterozygous. So I'm going to see heterozygous brown. Sorry, black. It's black, Nabi. Heterozygous black. Abi. Then 2. This is the second one we are seeing. Then we're now going to see what now? This one is still this one is still heterozygous black. So we are seeing, we cannot count them twice. We have seen what? Homozygous black, heterozygous black. Then this last one, we're now going to see what? Homozygous brown. Which is what? This BB, okay? This heterozygous black is what? Is BB, right? So this is what? Then this homozygous black is what? Is BB. So these are the 3 different genotypes we're going to see. We're going to see what? An homozygous black. We're going to see an heterozygous black. Then I'm going to see an homozygous what? Brown. So those are the 3 genotypes we will see. You will not count this one twice. You have counted this one, you have counted this one, it's appearing here again, so you not count it. Then you are seeing this, okay? So we are seeing 3 different genotypes, all right? Well, although the phenotype ratio will be 3:1, the genotype ratio here, genotype ratio, who will tell me? Genotype ratio will be what? Okay, obviously will not be what now? Will be 1, because this homozygous black, 1 to what? To 2, okay, heterozygous black, then to what? To 1. Homozygous brown, right? So the genotype we are going to see, the number of genotypes we are going to see here is what? Is 3 different types. You know, go be 4. You know, go say this one 1, 2, 3, 4. Okay, this one is appearing twice. We're not counting twice. Doesn't make sense. Okay, so this question now, anybody can easily fail it. Even a teacher of biology can fail this question, okay? Because of maybe overzealous, not because you don't know him, right? Because of overzealousness and because the person did not pay attention to details. So you must learn to pay attention to details, understand what the examiner is asking of you. Okay, that is why you need what? A good JAMB application like this, okay? So once you have the O3 Schools app, most of your problems are solved. O3 Schools is the way to school. Trust me, all right? So whether it's science or art, do not mess with that. Let us see if we can take one more. Okay, this is a 2008 question. 2008 question. This is a 2008 question, number 42. Okay, 2008, number 42. Okay, we're given a diagram. They say use the diagram to answer the question. They say which of the 4 offspring of the cross would be short if T is the gene for tallness, if T, the gene for tallness, is dominant to what? To T. Okay, so we're giving what? T. We're giving T. We're giving what? All right, so this is the table we are giving. All right, so something like this. Okay. All right, so then we now have what? T also. T. Okay, so now they say which, this T is dominant, obviously. This one is recessive. Okay, so they say which of the 4 offspring they are giving here as I, yes, what? As I, yes, I, yes, IV. So this is also a monohybrid cross, you know, right? So now, let us find our crosses. If we cross, we're going to have TT, all right? They say which of the 4 offspring of the cross will be short, okay? Obviously, this one is tall. This one is tall. This II is what? Is tall. This I is tall. This II, is it tall? Yes, it's tall. It's heterozygous tall. Then this III, is it tall? Yes, it is heterozygous tall. Then this IV, is it tall? No, it is short. Okay, so our answer will be IV. That is option what? Option D. Let me see if we can see one more, then we're done. And enter our Dihybrid Cross. They say a tall plant crossed with a dwarf plant. This is a 2011. Number 50. They said a tall plant crossed with a dwarf one and produces offspring of which half are tall and half are dwarf. Okay. Okay, so let's see this. 2011, number 50. All right. So now they say, what are the genotypes of the parents? Okay, what are the genotypes of the parent? Okay, a tall plant, they not tell us, this is where the examiners try to confuse you. Yeah, now they not tell us if it is what? If it is homozygous or heterozygous. They only told us what a tall plant. Okay, they only told us that a tall plant was crossed with a dwarf one, okay, and produces offspring of which half are tall and half are dwarf. Let us cancel the options. Let us remove some from the options. We will cancel. A said is TT and what? TT. If we cross this, what I want to have, this will cross with this TT. I mean, this will cross with this, okay? I'm going to have what now? TT also. All right, then this now will cross. So I'm keeping this capital letter T constant, and this will cross. Okay, cross. So I'm going to have TT. Then this and this will also cross. So I'm going to have TT. All right, this and this will cross. Okay, so now I'm going to have TT. Now, if you look very well, is this one tall? Yes. Is this one tall? Yes. Is this one short? Yes. Is this one short? Yes. Okay. They say, what are the genotypes of the parent? Okay. So since option A has already given us our answer, because we're told that half were tall and half were dwarf. Okay. Among the four, these are four offsprings. Okay. The first one here, this one is what? Is short. This one is what? Is short. This one is what? Is tall. This one is what? Is tall. All right. So obviously we are getting our answer. Half of them among the four, two are short. Okay. Two are what? Are tall. So, okay. So imagine, let's say A did not give us our answer. We'll have gone to B. We'll have checked B. B, look at the genotypes of what? Of B. TT and what? TT. Okay. So, sorry, no. T, capital letter T, capital letter T. Let's cross. TT. Okay. TT. All right. Then TT. Then TT. All right. So this one is tall. This one is tall. This one is tall. All of them are tall. So option B cannot be correct. The third one said TT and TT. Okay. So now let's cross. Obviously, this one I already know the result now. Okay. 3 will be tall. Yeah. 1 will be short. Okay. So it's still wrong. Okay. Because this is like our normal monohybrid cross. Now the fourth option said TT and TT. Obviously, this one, all of them will be tall, Abi. So you eliminate that one immediately. So our answer will be what? Option A. So let us just go to dihybrid cross. I'm seeing one more question here. Okay, you will answer those one by yourself. So let us go to dihybrid cross. Okay, dihybrid cross, so that we do not waste so much time. Please ensure you like this video. I've discovered that many of you do not support our videos. All right, you don't like, you don't share, you don't subscribe. Nothing does not concern you, which is very, very bad. Okay, it's bad not only because of you, but also because of your other students, your other students who are preparing for this same exam as you, who will need these videos very well. All right, please, even if you will not watch this video to the end, okay, just click on the like, just like the video. Click on the like. Only liking alone will send what will make this video to be suggested to them whenever they are on social media. Okay, whether you are watching from Facebook or TikTok or YouTube, please click on the like button. Follow us on TikTok and Facebook if you are watching from those channels. From YouTube, make sure you subscribe to this channel. Okay, subscribe to the channel. Check to see that you are subscribed. All right. Then, of course, very importantly, copy the link of the video from wherever you're watching for. Share it to your friends, share it to your loved ones, all right? And of course, God will bless you abundantly. So let us see dihybrid cross, okay? So now, in monohybrid cross, all right, we spoke about what? One character, okay? We spoke about what? One character. All right. We considered only one character. We said mono means what? One. All right. So also now, when it comes to, in fact, the dihybrid cross necessarily confuses students very well, even teachers. Trust me, it confuses even teachers. All right. So, but I'll try as much as possible to make it understandable for you. All right. Now, for dihybrid cross, now di, on the other hand, it means what? Two. Okay. So in monohybrid cross, which meant one, we consider one character. For example, we said what? Seed. Okay? Okay, no, not seed. We consider what? The height as a character. Okay? It was the height we considered, all right? But of course, since it was the pea plant that Mendel used, all right, there were so many contrasting characters. There was seed shape, pod color, okay, even flower position and the likes, okay? So among all of those contrasting characters, all right, we consider just only one character in monohybrid cross. But in dihybrid cross now, okay, which means what? Since di means two, it means here we're considering what? Two characters. we are not considering one character as in monohybrid cross, okay? That means we can consider, for example, height as a character, and also consider, for example, flower position, okay, which is another word contrasting character. Or we could consider pod color, okay, and pod shape, all right, which is another contrasting character. But for dihybrid, we must consider two characters the same time. Why for monohybrid we consider only one character, which is why when we're discussing monohybrid cross, we spoke only about height. Okay, height, we have two possible alleles with each other, they are tall or short, isn't it? Now that is that for that. But for dihybrid, we consider two characters. Are we clear now? So now let us begin. So for dihybrid cross, we have understood that we are considering two characters. What two characters are we considering? Right? We are going to consider seed color, okay, and then seed shape. Okay, so for dihybrid cross, we are considering two characters, seed color and seed shape. All right. So of course, definitely. Now for the color, all right, for the dominant trait, all right, we are going to consider yellow color for the dominant. Then for the seed shape, we are going to consider round for the dominant. Okay, so for the dominant, we are considering yellow and round. Are we clear? We are considering yellow seed and round seed shape. Now for the recessive traits, okay, we are considering green seed, okay, green seed color, and and wrinkled. Okay, so this is what we are considering when it comes to dihybrid cross. This means that that these ones here, they are the alternative alleles, okay, or alternative traits, okay, that we are considering. All right, so seed color may be yellow or green, okay, it may then the seed shape may be either round or wrinkled. So let us say we are considering the parental generation now, okay, all right, this seed color, this seed shape, okay, the parental generation, all right, definitely we are going to consider pure breed only, okay, we are considering pure breed, okay, pure breed, all right. So now yellow color will be YY, okay, round seed will be RR. Are we together? Now, Y for the green, okay, it will be now? It will be YY, all right, it will be YY, small letter showing that it is recessive, okay, then wrinkle will be now? Small r. Are we together? Okay, so for yellow and round, all right, I'm going to have YY. This is the phenotype, okay, the phenotype. This is what we are seeing, okay, it is yellow, okay, it is round, all right, it is green, it is it is wrinkled, okay, so here I can just say phenotype, all right, phenotype, all right, it is yellow, it is round, it is green, it is wrinkled. So then now, what are the gametes? Okay, sorry, this is not the phenotype. This is the one we are seeing is yellow and round. Now this is phenotype. Okay, this is the phenotype. Okay, yellow round. What will they see? Now be yellow. This one yellow. This one round. Now what will they see outside? Say this one green. This one they wrinkled. Okay, on the outside, this is what you are seeing. Why this one? Okay, that ones that are not visible to the eye. So this one is is the genotype. Okay, now we have our gametes. Okay, so now how do we get our gametes? Now, when we're discussing monohybrid cross, all right, we said we considered height, okay, TT and TT, okay. This is tall, this is dwarf, all right. And then what we said? We said we can break this into T and into T, okay. Then break this one into T and T, all right. This one will bring one allele. This one will bring the other allele. Okay, they'll combine. Okay, we now have T. All right. But when it comes to dihybrid cross, okay, we cannot do this. We cannot do something like this. We cannot say, okay, the gamete will now be Y Y, arrow, arrow. Okay, now Y, arrow, arrow. If you do something like this, it means that what we'll be considering what monohybrid cross, not dihybrid cross again. Okay, so when it comes to what monohybrid cross, okay, so when it comes to what dihybrid cross, what we simply do to get our gametes, all right, what we simply do, we take what one of these, and then we take what one of these, all right. So what we simply do, we're going to have Y and arrow together. Then here we're going to have what now? Y and arrow what together. This is what we do when it comes to what to dihybrid cross. So then now let's cross them. Okay. Now if we cross this, okay, if we cross this with what with this, what I'm going to have? If we cross, I'm going to have Y, okay, and what and Y. Then I'm going to have what R and what and R. Okay. So this is what our F1. Okay. So this is what we get in our what in our F1. Now this result of getting our F1, all right, is it, this is F1, is it homozygous or it is what? It's heterozygous. Okay, is it homozygous or heterozygous? Obviously, as you can see here, it is definitely what? Heterozygous. It is not homozygous because we're having what? The similar what alleles, all right? So we have what? Y, R. So now, what did Mendel do? So Mendel what? Self-pollinated what? This what? F1 again, okay? He what? He crossbred them, all right? So he pollinated this, and then what? What happened? Let us quickly see what happened next. All right, so this one was what was crossed. So now our, obviously this one, what are we seeing here? Okay, the phenotype, what we believe showing us, seeing us was yellow and round. Okay, despite it's what? Heterozygous yellow and round. This is what we are getting in the F1, all right? It is heterozygous, but it is definitely what? Yellow and what? And also what? Round. Okay, so we have Y, R, our F1. So now our F2, the parents, this now became what? The parent. So Y, R now became parents. Okay. They will also have what? Y, R. So it is now what? A parent. So now after what? Also what? Do a cross on it. This is where it now becomes very, very tricky. Okay. Becomes very tricky. Now, how do we get our gametes? How do we get our gametes? Okay. As we are here now, how are you going to get your gametes? Okay, so now I'll tell you how to do it in a very, very easy way, so that you do not have to stress yourself at all. Okay, so to get your gametes, simply bring this one out, Y, then R. All right, then just keep this one constant and what? I need to cross these ones. Okay, so now keep this Y constant, use this to cross all of these. Now this will cross this, so I'm going to have capital letter Y and what? Capital letter R, all right? Yellow and round, all right? Then use this one also to cross this. I'm going to have this, all right? Capital letter Y and what? And R, small letter R. Then use this small Y to cross this again. I'm going to have capital letter, okay? You're going to have small letter what? Y rather, and what? Capital letter what now? R, all right? Then, of course, they use this one also to cross this. If you cross this, I'm going to have this. Hope I got the cross. This is this and this. All right. Sorry, I didn't do that. So then they use this one to cross this one. I'm going to have this and this. So I'm going to have small letter Y and small letter R. All right. So these are our four gametes. Okay. These are our four gametes. And this is where the law of independent assortment. The law of independent assortment was postulated by what? By Mendel, all right? So now, what that law said, I will explain it in layman's terms. I'm going to look at law of independent assortment very, very soon, okay? After I do this small explanation. When we're crossing, all right, the alleles were assorted independently, all right? It does not mean that this one and this one must be together, or this one and this one must be together, okay? They were assorted independently, all right? They were not controlled, all right? They arrange themselves the way they want. It does not mean that this Y and this arrow must go together, or this Y and this small arrow must go together, or this small Y and this big arrow must what? They go together. You can see the way they what they will arrange, all right? They will arrange what? Independently, all right? I will explain the law of independent assortment the better way that you understand. But let's also forget our gametes, all right? So now we have gotten our gametes. So that means our gametes here will be what? Y, R, okay? Y, capital letter Y, and what? Capital letter R. They are going to have what? Y, R. They are going to have what? Y, R, all right? That means that what? These gametes also will be here. I'm going to get it here. Y, R, Y, R, Y, R, Y, R. So now we will be able to what? To get our what? Our gametes, okay? So now we're not going to what? Do the cross, okay? We're going to use our Punnett square to what? To do the cross, to make it very easy for us, okay? But before we do this cross, okay, let us keep our gametes in one place and talk about the law of what? Independent assortment, all right? So now let us go. Let's talk about the law of independent assortment, all right, in a more understandable form. Okay, so please ensure you have liked this video, you have subscribed to this channel, you are sharing the video link to your friends and to your loved ones. Okay, let all and sundry come, learn, succeed. That is our goal, that's our target. All right, we do not want you writing JAMB twice. Okay, so let us look at the law of independent assortment. Now, what does the law of independent assortment says? Okay, the law of independent assortment says that when two or more traits are inherited together, right? The genes for each trait separates. By separation, okay, they mean what? Assort, all right? When two or more traits are inherited together, the gene for each trait, they separate independently during gamete formation, all right? So that's what the law is saying. The law of independent assortment says that when two or more traits are inherited together, the gene for each trait will assort. They will separate on their own independently during gamete formation. This therefore means that what? That the inheritance of one trait does not affect the inheritance for another trait. Okay, Mendel gave the law of independent assortment. Okay, when he was working, okay, in the monastery with the what? With the pea plants. Okay, so when I was working with the pea plants, all right, he gave us this law of what? Independent assortment. So in general, just think of it like this: that when you are making gametes, okay, either what? Egg or sperm, each gamete will get only one copy. Each gamete will get only one copy of each gene, all right? Each gamete will get only one copy. When you're making gametes, all right, each gamete will get only what? One copy of each what? Gene. So therefore, the way one gene goes into the gamete, okay, does not affect the way another gene what will go what in, all right? So independently, all right, they assort independently. They assort, okay, independently. That's why the law of independent assortment is saying that what? When two or more traits are inherited together, the gene for each trait, they separate or they assort, usually during gamete what? formation. All right, they assort independently, and the inheritance of one trait does not affect the inheritance for another trait. But why does this happen? Okay, this happen because genes for different traits, okay, genes for different traits are located on different chromosomes. Okay, this is why this is what majorly what happens. Therefore, during what meiosis, okay, which is what cell division to form gametes, okay, this chromosome they separate randomly. They separate randomly, okay, so that what traits do not what influence each other, so that traits do not influence what each other. Okay, so they are what they go into the gamete what independently. Okay, independently. That is the law of what independent what assortment as given by what by John Gregor Mendel. For example, in humans, okay, eye color and hair color, they are what? They are different, are they not? Okay, somebody can have what? Let me say blue eyes, and at the same time have what? Brown hair. Abi, brown hair. Abi now, isn't it? Okay, someone can have blue eyes and have what? Brown hairs. Okay, so the trait for this, okay, the gene for this does not affect what? This one. They are independent of what? Of each other. They are independent of each other. Now let us quickly see the usefulness of what? Of the law of what? Independent what? Assortment. The usefulness. Okay, so we are talking about this law now, but why is it what useful? It is useful for so many reasons, okay? In the agricultural field, okay, in genetics, okay, and so many other areas. So let us just list some of them, all right? Because you do not know where JAMB will strike you from, or WAEC, or NABTEB, or NECO, all right? So please ensure you are armed to the teeth, all right? So let us see the usefulness. Now, one of the usefulness of what the law of independent assortment is in agriculture and animal breeding. Okay, so number one, in agriculture and animal breeding. Okay, now law of independent assortment helps what farmers. Okay, it helps farmers. It helps what scientists. And farmers, okay, to combine desirable traits, okay, to combine desirable traits, okay, in both crops and animals, all right? So law of independent assortment is useful in agriculture and animal breeding. It helps scientists to or farmers to combine desirable traits in crops and animals. All right. For example, a maize, you know maize now, right? It's not every maize that is sweet, okay? Some maize, even if you just pluck them today, if you know sweet like that. Okay, the only thing that will make it maybe sweet, normally you know that fresh corn is usually sweet, but some maize are definitely sweeter than others. All right, so now a farmer can combine a breed of maize that is sweet, and some maizes that they are very susceptible to diseases. Okay, so a farmer can combine a sweet maize plus a maize that is not prone, okay? That is not prone to diseases, okay? So this one is one trait, this one is another trait, okay? So it's sweet, okay? And it is not susceptible to disease. So you can now interbreed them together, all right? So to now give better maize, all right? Give us a better quality of maize, all right? That is sweet and that is not prone to disease, okay? So it helps agriculturists, it helps our scientists, okay? It's very useful in animal breeding likewise, all right? So now, I think I saw somewhere that you can interbreed a horse and a donkey. Okay, I think to give you what a mule. All right. So if you interbreed them, all right, it's not no donkey and horse usually for transportation, basically. Okay, this mule is even more effective for transport. All right, so it's also very, very useful. All right. So now let us go. Now, number 2, like I said, it's useful for what? For genetic what research. All right, for genetic research. Number 2, useful for what? For genetic research. This one, it helps scientists also. Helps scientists map genes on chromosomes, okay, and understand genetic disorder and understand genetic disorder. All right. So also the law of independent assortment helps in genetic research. Okay. So it helps scientists to map genes on chromosomes and understand what several genetic disorder. All right. We'll look at sex-linked character soon. Okay. Okay, we want to talk about applications of what of heredity. Okay, we're going to see what a lot of what genetic disorders. All right. So just know that what law of independent assortment is very, very useful here. Okay, helps scientists to map genes on chromosomes and understand genetic disorder. Also, it is useful in medicine. Medicine, all right. So it is used to what? To study. Used to study how different traits and diseases are inherited separately or together. All right. So it's also very useful in what? In medicine. Just to study how different traits and diseases are inherited either separately or together. Okay. So in medical field also, the law of independent assortment is very, very useful. Used to study how different traits and diseases are inherited separately or together. Then number 4 also, evolution. Okay, so evolution over time increases what? Chances for what? For survival. So the law of what? Independent what? Assortment, okay, helps what? In evolution. It helps us to understand variation in offsprings. Helps to understand variation in offsprings. Okay, so that is another very, very important usefulness of the law of independent assortment. Then number 5, we have forensic science. Forensic science. So for forensic science, it helps in DNA analysis. Okay, in DNA analysis. And why does it help in DNA analysis? Because each trait, they assort differently. They assort what? Independently. Okay, or what? Differently. All right, you have heard before that when they will say that what? Everybody's DNA is what is different from the next person. Okay, so my DNA is different from your own. Okay, so in forensic science, it helps what? In DNA analysis, because for those traits, they are assorted, they are separated, okay, independently, making each person's what? DNA unique to the person. Okay, so these are some of the usefulness of the law of independent assortment. All right, so now let us go back to our cross in the Dihybrid cross, and let us finish the Dihybrid cross. All right, in the next class, we're going to discuss sex determination. All right, so how is sex determined? You are a male, you are a female. Okay, so how does that come about? How does it happen? Okay, we'll understand it better in the next class when we talk about sex determination. All right, so let us round up the Dihybrid cross. So in our first, in our F1, okay, we had what? Y, and what? R, isn't it? Then we got our what? Our gametes. Okay, this is our F1. Okay, so it is this one we are crossing with what? With this one. Remember where we stopped right before we started talking about law of independent assortment. Okay, so this is where we are. Now, we found successfully, we find our gametes, okay? How did you find our gametes? We said Y, Y, R, R, isn't it? We use this to cross this, all right, to give us YR, okay? So we have what YR is one of the gametes. Then we use this also to cross this, to give us Y and R, capital letter Y now, and small r. Then this one is already done. Now, keep this one constant, use this one. So this times this, okay? So this times what? This. Have small y and what? Big R. So have small y and big R. Then we'll now have what? The next one is what? Okay, so this one is still constant. We use this one to cross this. So I'm going to have this and this, which is small y and small r, okay? Small y and small r. So this is how we got our gametes, okay? And you can see that all of them, they are assorted separately and independently. All right, so that means also here, the same gametes are going to have now because we're crossing this with this, the same thing we're crossing. All this, our result in the F1 now became the parent in F2, okay? So now I'm going to have Y, R, Y, R, Y, R, Y, R, all right? So now, we use our Punnett square to get what? Our result, okay? For what? Our cross, okay? In the what? In the F2. Okay, so let us get our Punnett square. Okay, this will be big. Okay, so now we have a lot of different combinations. We have a lot of different combinations. All right. So now I'm going to have YR. Okay. Okay, we are going to have what? YR, capital letter Y and small letter R. We're going to have what? YR. Then also I'm going to have what now? YR. Okay, so up here also, up here, I'm going to have what now? YR, all right? Also, so for this other parent now, then also I'm going to have YR. I'm going to have what? YR. Is this YR? Okay, I think it's Y and capital letter R, sorry. All right, this is Y, capital letter Y, small letter R. Then this one, small letter Y, capital letter R. Do not make the mistake, please. Okay, so I'm going to have what? Y and R. This is the third one. Okay. We are going to have the last one, which is what now? Small letter Y. Okay. Small letter what? R. Okay. So now this is what we got. Now we begin what? Our crossing. What we've crossed YR times what? YR. What I'm going to have? I'm going to have what now? Y, Y, R, R. Abi? Yes. So this times this also. I'm going to have what now? Y, Y, arrow, arrow. All right. Then this, we multiply this. I'm going to have what now? Y, Y, Y also. Then I'm going to have what? Arrow, arrow. Isn't it? Then this, we multiply this also. I'm going to have Y, Y. Okay. Arrow, arrow. All right. So now I've gotten that first what line. Now let us go to the next one. We used to multiply this now. This is capital letter Y, right? Yes. So now capital letter Y times this, Y, Y, arrow, arrow. Okay. This will multiply Y, Y. Okay. Arrow, arrow. You should multiply this. Y, Y, arrow, arrow. This will multiply this. I'm going to have Y, small letter Y, small letter arrow, small letter R. Okay. So let's go with this one now. We used to multiply this. I'm going to have Y, Y, okay, Y, Y. I'm going to have arrow, arrow. This will multiply this. I'm going to have Y, Y, okay, arrow, arrow. Isn't it? Okay, this will multiply this also. I'm going to have Y, Y, arrow, arrow. Then this will multiply this. I'm going to have Y, Y, capital letter arrow, small letter R. All together. Then this also will multiply this. I'm going to have small letter Y, capital letter Y, capital letter arrow, small letter R. All together. So this will multiply this also. I'm going to have small letter Y, capital letter Y, okay, small letter arrow, small letter arrow. So this will multiply this also. I'm going to have what now? Small letter Y, small letter Y, okay, capital letter arrow, small letter R. So this will multiply this. I'm going to have what now? Small letter Y, small letter Y, small letter arrow, small letter arrow. Now, at the end of what? Of our F2, how many different combinations do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, so we have a total of 16 possible combinations. Okay, after our what? Our F2. All right, so this is the result, okay, of our what? Of our cross in the F2. Now, but what is the result? We need to what? Find our what? Our phenotype ratio. Now, to get the genotype ratio is very, very complex. Very, very complex. So we're not going to talk about what the genotype what ratio. Okay, we can actually do it, but though it's complex, and we don't really need it. Okay, so but of course, what is very important is what the phenotype ratio. We must know the phenotype ratio at the end. Okay, so now we have what? Yellow and round seeds, isn't it? We have yellow and then round seed. How many yellow and round seeds do we have? Okay, let us look at them. This one is yellow and this one is round. Okay, so this is one yellow and round seed. This one is what? Is yellow. This one is also what? Round, isn't it? Why is it round? Because this one cannot what? Overshadow this what? R. This small R cannot overshadow this big R. Right? So if this small R is recessive, this one is dominant. Okay, so we have what? Another what? Yellow and round. Is this one yellow and round? Yes, it's yellow and round because this small Y also cannot overshadow this big Y. Can it? No. Okay, so this one is round, obviously. So we have another yellow and round. Now, is this one yellow and round? Yes, it's still yellow and round, isn't it? Because what? We have what? The Y, the capital letter Y is dominant over the small letter Y. The capital letter R is also dominant over the small letter R. So it's still yellow and what? Round. That we have seen 4, isn't it? Is this yellow and round? Yes, it is yellow and round. So this is 5. Is this yellow and round? Okay. No, this is not yellow and round. This is what? Yellow and wrinkled. Okay. So we have found what? Yellow. Let us leave this one. Okay. So now, is this yellow and round? Is it? Yes, it is yellow and it is round. Okay. So yes, it's another yellow and round. Now, is this yellow and round? No, it is yellow. Okay, but it is what? Wrinkled. That's not what we're looking for. Now, we're looking at yellow and round now. Yellow and round first. Okay. So now, yeah, is this yellow and round? It is yellow and round. The Y is dominant over the Y, over the small Y. So yes, it's yellow and round. Is this yellow and round? Yes, it is yellow and round. Okay. Now, is this yellow and round? No, this is what? Green and what? And round. Green and round. Okay. Green and round. This one is what? Green and also round. This one is what? It's yellow and round, all right? Why is it yellow and round? Because we have what? This capital letter Y is dominant over this small letter Y. Okay, so it is yellow and round. Is this yellow and round? No, it is yellow. Yes, it is yellow, but it is not what? Round. It is wrinkled. This yellow and round? No, it is green and round. This yellow and round? No, it is yellow. Sorry, it is green and what? And wrinkled. Okay, so now we have how many yellow and round? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so we have what? 9 yellow and round what? Seeds. So now let us look at round and green. Round and green. Okay, so let us find the one that is round. Okay, and it's green. So green and round. Now let us go. So this one's obviously out. Okay, we have anyone that we have marked here. Okay, is out. So green, round and green. This one is yellow. Okay, it is wrinkled. Okay, so it's yellow and wrinkled. Okay, so let me just say add. Okay, let us finish with round and green. Okay, so now this is so yellow and wrinkled. Abi, no, obviously we are done with this one. So this one is also what? Yellow and wrinkled. This is what? Yellow. Okay, and round. Oh, sorry. This also, no. We are done with this. Now, this is what? Green and round. Abi, we're looking at green and round. So we have found one. Let's asterisk it. Okay, green and round. Is this one green? Yes. Is it round? Yes. Okay, because this small arrow is recessive. This arrow, capital arrow, is dominant. Okay, so this one is green and round also. So we have found 2. Green and round again. Let's go. Is this green? No, this is yellow. So is this green? No, this is still what's yellow because what? This Y, capital letter Y, is dominant over this small letter Y. Is this green around? Yes. This one is what? It's recessive, this is dominant. So it's green and round. So we'll find another green and round. Is this green and round? No, this is what? Yellow. So this was green and wrinkled. So how many green and round did we see? Round and green. Okay, 1, 2, 3. Okay, so how about 3? Okay, so now I'll be able to successfully find yellow and round, and then I've seen round and green. Okay, yellow and round is 9. We'll find 9 combinations. Round and green, we'll find what? 3 combinations. So now let us look for wrinkled and yellow. Okay, wrinkled and yellow. All right, wrinkled and yellow. All right, so we're done with this. No, this makes our work easy. Okay, wrinkled and yellow. This one yellow, yes. Is this wrinkled? Yes. Okay, all right. It is yellow. It is what? Wrinkled. So let us use O to represent that one. This is what? Wrinkled. It is yellow, and it is what? It is wrinkled. This one we have used this before. Now this one, is this yellow? Yes, it's yellow because Y is dominant over this small y. This capital letter Y is dominant over this small letter y. But is it wrinkled? Yes, it is wrinkled. Okay, so we'll put that sign. Let's go. We have used this one, 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 we have used this one. This one, yellow and wrinkled. Is it yellow? Yes. Is it wrinkled? Yes. Okay, I hope I'm clear. It is capital letter Y and small letter y, so it's also what? Wrinkled. All right, so we have what? Another wrinkled and yellow. Then is this, we have used this one. This one is what? Is green and wrinkled. I'm not looking at green and wrinkled. Okay, so now how many wrinkled and yellow do we find? 1, 2, 3. Okay, so how about 3? Okay, so 3 yellow and wrinkled. Now let's look at wrinkled and green. All right, we have used this one before. We have used this one. 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 You have used this one, 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 only this one that's many. Okay, so we'll have only one green and wrinkled. Okay, it's wrinkled and it is what? It is green. All right, this shows that that at the end of of our dihybrid cross, okay, our phenotype ratio, okay, is what? Is 9 ratio what? 3 ratio 3 ratio 1. Okay, so that is what our phenotype ratio at the end of of dihybrid cross. Okay, at the end of dihybrid cross, that is what our phenotype ratio. 9 ratio 3 ratio 3 ratio. It is very important. You must take note of that. All right, the phenotype ratio at the end of our dihybrid cross is what? 9 ratio 3 ratio 3 ratio 1. Okay, now like I said, the genotype ratio, okay, is very complicated. We can decide to do it too, but it's actually not needed in this class. All right, so we are not going to touch on that. Just know what the phenotype ratio. Then for the monohybrid cross, know the genotype and the phenotype ratio. Okay, we say the phenotype is 3 ratio 1, the genotype is 1 ratio 2 ratio 1 in monohybrid cross. While in dihybrid, okay, phenotype ratio is 9 ratio 3 ratio 3 ratio 1. Make sure you do this table by yourself. Okay, try and do it by yourself and understand what we did here today. It's not something difficult. It's not something you cannot do. You can do it. Okay, do it by yourself. Find the phenotype ratio. Okay, that will be your assignment. So this is where we're going to what? Is there another thing I want to add? Now, there are some amendments to Mendel's law, okay? Okay, let's quickly see some of what this was amendment, okay? There are some terms we have to get used to here. Number one is incomplete dominance. Incomplete dominance. Please like this video, subscribe to this channel, share, copy the link of this video and share to your friends and loved ones. Download the O3 Schools app, okay, and start studying towards your success. It is very, very important. If it were not important, I would have told you so. So let's look at the concept of what? Incomplete dominance. All right, now for incomplete dominance, all right, normally we know that in Mendelian genetics, according to Mendelian genetics, okay, it says that what? If you have a round seed, okay, a round seed, and then a wrinkled seed, okay, let me say round seed is what is R, it is dominant over the wrinkled seed, which is what R, isn't it? Now, if you have what? A round seed together with a wrinkled seed, what happened? All right, the round, which is dominant, okay, will what? Will overshadow what? This recessive. Okay, if they come together, I'm going to have something like this, isn't it? RR. Now, what will happen? Will this seed be round or wrinkled? It will be round. We say it will be round, right? Why? Because this round seed is dominant over the wrinkled seed. This is what Mendelian genetics told us, Abi. Now, but for incomplete dominance, neither alleles. Let me put it down for you to see so that you can understand better. Neither alleles is completely dominant. Okay, so in incomplete dominance, right, neither alleles, okay, not this or this is what? Completely dominant. This one is not completely dominant over this. This one is not completely dominant over this, okay? So neither alleles is what? Is completely dominant when it comes to what? Incomplete dominance. What do you observe? The two alleles, instead, they blend together, okay? Blend their effects in the heterozygous condition. In the heterozygous condition, all right? So for incomplete dominance, neither alleles is completely dominant, okay? The two alleles involved, they blend their effects in the heterozygous condition, making them to produce a trait, okay? Making them produce a trait that is in between. Okay? So that is what incomplete dominance talks about. Neither alleles is completely dominant, okay? Not this one is completely dominant. Instead, they blend their effects, okay, in the heterozygous condition. This is heterozygous, right? Because they are dissimilar alleles, all right? But they blend their effects in the heterozygous condition, producing a trait that is in between. Then what do you observe? The heterozygous offspring now, okay, it now shows what? An intermediate trait. Intermediate trait, okay, that is different from what? From both parents, okay? So the trait that it will now show is not seen here, neither is it seen here. Now, let me just show you a good way to remember incomplete dominance. It's very easy, and it comes out in your exam, all right? So please ensure you pay good attention to it. If it was not important, I will not have be saying it at all. Please ensure you pay cognizance to it. For example, you know when you mix, those of you that know about colors very well, if you mix red and white together, okay, let's say we have red, which is arrow. Let us say we have white, which is what? RR. If you mix them together, what do you get? Okay, you get what? Pink. Do you know that when you mix red and white together, you get pink? Yes, okay, so that is what incomplete dominance is, all right? So normally, you are supposed to have what? Arrow arrow, okay? Arrow and what? An arrow, isn't it? If one brings one allele here, this one brings one allele, all right? So we're going to have arrow, arrow. Is it normally, and this red is supposed to dominate. But in incomplete dominance, what happens is that what? As this red and this white are coming together, okay? Different alleles, but they blend together to give you something different. Okay, this one is this is one we call the intermediate trait now. In the intermediate trait that is not present in the parent. It's not here. We are seeing what? White. Okay, so that is the concept of what? Of incomplete or dominant. Neither allele is completely or dominant over the other. The two alleles, they blend their effect, okay, in the heterozygote condition, producing a trait, okay, that is what? That is in between, okay, that is in between, okay. We see this in a lot of, an example of what? Of organisms, we see this, one we call the snapdragon flower, okay, we see this in the snapdragon flower. We see this also in the Andalusian fowl, okay? We see this in the Andalusian fowl, okay? Also, we see this in the 4 o'clock plant. 4 o'clock plant. Okay? And also some cattle. Some cattle also, we see this, all right? Red and white cattle, they come together to now form a new color called cattle, some cattle. Okay, some cattle, okay, a red and white cow will come together and now form a new intermediate form, completely called what? The Rowan, R-O-A-N, right? So that's also an example, okay, of organism that shows what? This incomplete dominance. And also in incomplete dominance, the genotype ratio, please take note that the genotype ratio, okay, and also the phenotype ratio, they are what? They are the same since their effects what? Blends. Okay, so the genotype ratio and the phenotype ratio are the same. Now let us talk about codominance. All right. So please, you have no reason not to like this video. Please like this video, subscribe, share. Okay, download the app, activate it, pay. Activate it. Tell your friends about the app, okay? Tell your friends about the app. It will help them a lot. Okay, let us look at codominance, all right? Let me just put a space here. Codominance. Now, in codominance, okay, according to Mendelian genetics, we already saw, okay, that the dominant trait, okay, will always what? Will overshadow the recessive trait, the recessive trait, okay? This is what Mendelian genetics told us, isn't it? Now, but for codominance, okay, just like what we saw in incomplete dominance just now, we saw that the effects we blend together, okay? But in codominance, these alleles in codominance are equally strong. Both alleles are equally strong, and they are expressed together, and they are expressed together in the heterozygous condition. All right, so neither one hides the effect of the other. Okay, so they don't hide the effect of each other. They don't blend together, just like when we saw red and white turning to pink just now. Okay, no, they don't blend together. All right, so now, for example, in Rowan cattle, which is an example of Rowan cattle. Okay, it's an example of codominance, okay, of codominance. All right, so now what happens is that you interbreed a red cattle, okay, with a cattle with red color with a cattle with white color, okay, and when you now see the cattle, you see red, you see white, okay, fully expressed, okay, on the body of the cattle, okay, of the offspring. All right, so that is now codominance is different from incomplete dominance. In incomplete dominance, the alleles, okay, they blend together, okay, and then they now form an intermediate trait. All right, but for codominance, they don't blend together. All right, both alleles, they show their full force, okay, they show their self what in full effect. Example, like we see in this Rwanda cartoon, also see it in the human ABO blood group. Okay, I will have explained it. Okay, but I don't think there's any need. Okay, we also have another one called the speckled flowers. Speckled flowers. Okay, so another example of what a species that what we will find what codominance. All right. So now let us look at the next term, which is for linkage. Linkage. Please like this video, subscribe, share, like, subscribe, and share. Like, download the O3 Schools app, I dey sing now. Okay, so let us look at what linkage. Now to linkage, and according to the law of independent assortment, okay, that we saw, okay, we said that different traits, they assort what independently. That's what we learned from the law of independent assortment, isn't it? Now, linkage is different, okay? Now, linkage is telling you here that that the genes that are located on the same chromosome, okay, are inherited together. Okay, that's what linkage is telling us. Okay, that the genes that are located on the same chromosomes, okay, will be inherited together. Why would they be inherited together? Because they are physically connected. Because they are physically connected, okay? It means that if two genes are on the same chromosome, okay, they will be They will be inherited together, okay? If two genes are present on the same chromosome, they will be inherited together. That is what linkage is, okay? The genes that are located on the same chromosome are inherited together. They will not assort independently, okay? They will not separate, okay? They will flow into the gametes, okay, together and be inherited. All right, that's what happens in linkage. Just imagine as if maybe there are two people on the bus. Two people now on the bus. Now, if you're on a bus to Lagos, okay, it's not the same destination you get to. Yes. So if two of you are on the same bus, if you are going to Lagos, okay, if the bus is going to Lagos, let's assume, okay, the bus is going to Lagos. That is the destination of the bus. All right. So nobody will come down along the road. Will both of you not get to Lagos the same time? You get to the same destination at the same time. All right. So if two genes are located on on the same chromosome, all right, what happens is that they will be inherited together. That is what linkage is talking about. They travel together into the same gametes. Okay, they travel together into the same gamete. All right, so that is what linkage is. The genes that are located on the same chromosomes are inherited together because they are physically connected. All right, so that is what linkage. So now let us look at polygenic inheritance. All right, polygenic inheritance. Polygenic inheritance. Now, normally in Mendel's genetics, okay, we know that a single gene is what controls a trait, okay? Just like we saw in Mendel's experiment on the peas, okay? Now, this is what happens. A single gene controls a trait, but in polygenic inheritance, this is not what happens, okay? Like, for example, in your family now, all right, if everybody in your family, if you came from a family where everybody is tall, okay, everybody is tall, your grandparents, your grandparents, all of them, they were tall people, okay? It is very, very, very most likely that you yourself will be tall. You can never be short, okay? So now, it's possible that maybe in your lineage, or some people were maybe medium tall, some people were short, okay? But along the line, okay, they begin to mix up. Even you said maybe you marry somebody that is short. okay? But your family, okay, because of that tall gene that is there, all of them will come together, they will blend to produce an effect of tallness. Okay, if you have, for example, if a father is what dark in complexion, the mother is dark in complexion, they most likely give birth to a child that's not fair, isn't it? That's what dark in complexion. That's what basically what happens. Okay, so that is what polygenic what inheritance. If in your family lineage everybody is dark, you say you are dark, you now marry a dark person, where fair one for come out. Okay, so naturally that baby is not supposed to be able to be fair. All right, so all of the effects, okay, all of those effects that come together, all right, to now give what a total package, what we call what this polygenic what inheritance. So you can say that what in polygenic inheritance, comma, a single trait is controlled by two or more genes. Two or more genes. Okay, so in polygenic inheritance, a single trait, okay, is not what controlled by one single gene. Okay, is controlled by what two or more genes working together and their effects add up to produce the final characteristic, to produce the final characteristics. Okay, so that is what polygenic inheritance. A single trait is controlled by two or more genes. They work together and their effects add up to produce the final characteristics. Examples like I talk about our height. Okay, height in the family. Okay, human height. All right, even eye color, skin color, eye color, right? Weight, okay, intelligence. Okay, all of these are what are examples of polygenic inheritance. Even what yield, okay, yield in crops. Okay, it's another what example. Milk production in cows, right? Milk production in cows. Okay, it's also what an example of what of polygenic what inheritance. All right, so please take note of all of that. So a single trait is controlled by two or more genes that are working together and their effect add up to produce a final characteristics. Do not confuse it. All right, now let us look at crossbreeding. Crossbreeding, please download the O3 Schools app. Or tell your friends, most importantly, tell your friends about the app. All right, nobody you they suppose pass. Okay, please. Let everybody know about the app, art or science. Okay, let them all get the app. Okay, let's look at crossbreeding. Now, crossbreeding involves of mating, all right? Mating animals of two different breeds, of two different breeds. There are two different breeds, but they are of the same species. The same species, okay, to produce offspring that have desirable traits, to produce offsprings that have what we call desirable traits. That's why it is used in animal husbandry, okay? Produce offspring that have what desirable traits from both parents. Okay, so that is what crossbreeding, mating animals of two different breeds, but of the same species, to produce offsprings that have desirable traits, okay, from what? From both what parents, all right? So that's what is known as what are crossbreeding. For example, you can have a breed A, for example, that is good. Let's say a cow now, good in producing milk. Okay, we can have another breed B. This one is what? Very resistant. You know, they quick sick. Okay, resistant to what? To diseases. Okay, so we now have what? A new breed that is what? Good in producing milk and also that is resistant to diseases. This is why crossbreeding is used very well, effectively, in what? In animal what? Husbandry to have what? Better yield. Okay, to have what? To produce what? Yeast that are what? Resistant to what? To diseases. Okay, so that is what? Crossbreeding. We have what? Three different types of what? Crossbreeding. There's one we call outcrossing. Outcrossing, number one, we have what? Outcrossing. All right. And in outcrossing, two animals, but of the same what breed. Animals of the same breed are mated. Okay, two animals of the same breed. Okay, all right, but they are what? Unrelated. All right, they are what? They are mated. Those are what? That is what we call outcrossing. When two animals of the same breed, okay, which are what? Unrelated. Which are what? Unrelated. They know they related. All right. Two animals of the same breed, okay, are mated what together. And the reason is to avoid inbreeding problems, to avoid inbreeding problems, and what? And to improve performance. Okay, I don't know if you have heard before they say dog. Let's say your dog give birth to the children. Okay, they say dog, no, they know whether I'm mama, but it is very, very bad, very, very wrong for you to use the offspring of that dog to go and mate the same what mother. Okay, it's very, very wrong. Okay, they say usually in local parlance they say you go born rubbish. All right, so to avoid what? Inbreeding problems, okay, and to improve performance, okay, outcrossing is what? Is very, 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 very good. Okay, so two animals of the same breed, but they are what? They are unrelated. That is the key word. Okay, they are unrelated. All right, they are not from the same what? Family tree. All right, so that is that. Okay, to avoid inbreeding problems and to improve performance. Then number two, we have interspecific cross. Interspecific cross. Now, in interspecific cross, two different species are mated. Okay, when they are mated, they give you something that we call what? Hybrid. All right, for example, like the one I told you, when a donkey, okay, is what? Crossed with what? With a horse. They'll give you what to call what? A mule. This mule, this one is for transport, this one is also for transport, but this mule, is strong past what both of them combine. All right, so they have what? Better what breed. All right, mule that is what? Specific for the one for a purpose. Okay, that's why they are calling it interspecific. It is specific for what? For transport. Okay, it can handle more than what a horse can handle on its own, or what a donkey can handle on its own. All right, it's called a mule. When a donkey and a horse are mated together to give what to call what? A mule. Then also we have what to call outbreeding. Outbreeding. Two animals of different species. Number three, outbreeding. Here we have what? Two animals of different breeds, but of the same species. Okay, when they are mated together, okay, that's what we call and know as what? As outbreeding. Okay, when two animals of different breeds but of the same species are mated together. Now let's look at the last one, which is what? Mutation. Let's look at mutation. Okay, after this we end the class. All right, sorry for taking a lot of time. All right, we have to just cross the T's and dot the I's according to the white man. Okay, so let's look at what? Mutation. Now we know that normally that our genes and chromosomes, okay, they carry what is known as as instructions. All right, for making what we know as as proteins. Apart from making proteins, they are also there for what? For controlling what we call traits. Okay, our genes and our chromosomes, they carry instructions that are useful for what? For controlling proteins. All right, so for producing proteins and also for controlling for controlling traits. But sometimes a sudden change happens in these instructions. Okay, like I told you, the codes, okay, the letters in what? In your DNA, A, T, G, C, okay? All right, adenine, thymine, guanine, and what? And cytosine. I say A pairs with T, G pairs with C. Sometimes it may be an error, okay, in this word code. Okay, maybe one letter will come before the other, and and so on and so forth, okay? Something like that can happen. A sudden change can happen in the set of what instructions, and when it happens, the effects sometimes are what are good, some are good, okay, some are what are bad, some are what are neutral. All right, but what we are very sure of is that what that it leads to what we call what a new word trait. It leads to what to a new word trait. Okay, so that is what we call what mutation. All right, it's a sudden permanent change. Okay, a sudden permanent change in what? In our gene. All right. So let me just put the full definition on what? On the board. All right. So mutation, you say, is a sudden permanent change in the DNA, a sudden permanent change in the DNA, okay, or chromosome that alters genetic information. A sudden permanent change in the DNA or chromosome that alters genetic information. All right, like we said, this change, okay, maybe maybe good, some may be good, some may be bad, okay, all right, some may be neutral, all right. So sometimes it will lead to to evolution. So many reasons, okay, but mutation is the only process that can alter our DNA. And we have different types of mutations. We have our gene. We have gene mutation. Okay, we have one we call chromosome mutation. Okay, we have genome mutation. Okay, so these are all types of mutation, and some mutation they happen naturally. Okay, they happen what we call naturally. All right, while some others are they are induced. Okay, maybe by overexposure to X-ray, okay, and what have you, all right, can lead to to induce mutation. And examples of mutation include one we call the sickle cell anemia. Okay, anemia. We have also albinism. We have one we call Down syndrome. Okay, we also have what is one we call blue eyes. Okay, all of these they are examples of mutation. So this is where we're going to stop today. All right, thank you for joining in. I've taken a lot of time. All right, I'm sure you enjoyed the class. All right, sure you understood what's dihybrid cross in its entirety. All right, I wish you all the best. I'll see you in the next class.